All right, good morning, everyone. We've got a packed schedule this morning, so we want to start right on time. It's 8:30, and we have an amazing lineup of speakers today for the interorganismal interaction session. I'm Susanna Tringe. I'll be the chair this morning. Just quick, the safety announcement for those of you who weren't here yesterday. If there is any kind of emergency, you can leave either way, go out and go either left or right, and just take the stairs down and leave the building. So this morning, our first speaker I'm excited to announce is Mary Firestone from just across the bay at UC Berkeley. She is a world expert in the soil microbial ecology. She did her PhD at Michigan State University and has actually been at UC Berkeley for the rest of her career and really has founded the areas of this field here. She has a long list of honors, including being a member of the National Academy. She's a fellow of the American Geophysical Union, the Ecological Society of America, the American Academy of Microbiology, and the Soil Science Society of America. And she's going to tell us today about her work studying the role of microbes in processing carbon and nitrogen in soil. So give a hand to welcome Mary.
Thank you, Susanna. And thank you for the invitation to have the opportunity to come talk to you all. Now let's see if this works. It does. I'm home, okay. So today, this session, I was invited to talk about interactions, and that is probably right now my favorite topic and what my lab is working most energetically on. And so we consider it a web of interactions. And we all know that over the years, there has been quite a bit of research on bacterial interactions, plant bacterial interactions, and I'm not going to go through the whole list, but they were two-part interactions. We're looking at how one type of organism affected the other. And we're trying to kind of mix that up and make it more complex and more realistic because we believe that interactions within domains and across domains are critical controllers of both the composition of the soil microbiome and the function of the soil microbiome. We cannot, we believe, effectively understand the interactions in isolation, that is bacteria, plants, without recognizing the other soil organisms and how they are impacting and affecting these interactions. We think it's the time to do these. We have the tools that we believe enable this exploration of ridiculously complex interactions. The molecular tools, the omics toolboxes, the isotopic tools. Today, I think I'm talking only about C13, but we also do a lot of work with O18 and N15, coupling those isotopic tools to the molecular tools. Spatially explicit mass spec, specifically, I'll talk a little bit about nanosims. Spatially explicit spectroscopy, which I don't think I'm presenting any of that data. And then mathematical modeling tools. And today I think I'm including information on what we call co-occurrence networks, that is network analysis. So I will proceed. The inhabitants, the bacteria, the fungi, the fauna, and the viruses interact. As we said, those are important controls on community composition, particularly in the rhizosphere, the zone around plant roots, and in the detritosphere, the zone around decomposing plant debris or plant litter. But also, these interactions are important controls on the flow and fate of plant root carbon as it enters soil. And we care about that last goal, what happens to the root carbon as it enters soil, because the carbon stock in soil is substantially greater than the carbon stock in the atmosphere plus land plants combined. So that which controls the stabilization, the input into stabilization in carbon, as well as decomposition and carbon leaving the soil as CO2 and methane, are critically important in the global carbon cycle. It's currently believed that these organisms in soil, particularly bacteria, fungi, viruses, and perhaps fauna, are determinants, are critical determinants of what happens to the carbon, whether the carbon coming in as exudates and root debris, is stabilized by interactions with soil minerals, or is stabilized by occlusion within soil aggregates. So we will explore most of those arrows, a few, with the ultimate goal of putting it all together. And I'm not going to be able to do that today, but I'll give you a progress report. Yes. Actually, the first time I used this slide, it was absolutely intended as a joke. We're trying, and people didn't laugh. They took me seriously. So I forget that it was originally that the concept that we would put all of those together. But we're making progress on some of the arrows and interactions. Our plants, our pet plants, are two common California annual grasses. Avena barbata, which is the dominant annual grass in Northern California, and Avena fatua, which is the dominant annual grass in Southern California. So our general approach will be, we will use stable isotopes to find the root carbon consumers, to map the pathways of where that carbon is going into soil. We will use isotope-enabled, genome-centric metagenomics to identify the members consuming that carbon, their metabolic characteristics or capacities, and the carbon sources they are using. We'll then use some metatranscriptomics to explore RNA-based viruses, as well as a portion of the eukaryotes that are also active in this food web using RNA-seq. Then we'll use that data for network analysis to attempt to begin to map the interactions that are critical. So we're focusing on the roles played by phage and viruses more generally, how fauna within the food web impact populations as well as carbon flow, and how a subset of the fungal population, that is arbuscular mycorrhizal fungi, AMF, interact with bacteria and fungi to mediate the flux and fate of root carbon. So we have, traditionally, over the last 12 to 15 years, started by 13CO2 labeling our plants in a greenhouse, in chambers. That's shown right here. But last year, we started 13CO2 labeling our plants in field sites, and that's at a place about 2 hours north of here, Hopland Research. It's a UC research station, and this is what our field labeling system looked like on an active day in April a year ago. I have a crew there now, and harvesting and separating the soils for a 16-day labeling that we just completed. These are the people who have made this possible. So you can see it's the layout. You can see the building in the middle looking down from above. That's the brain of the labeling chambers. There are 16 plots, 8 replicates of 2 different rainfall patterns, a normal rainfall pattern, 50-year average normal, and a half normal rainfall pattern. So we're trying to look at the effects of drought, and I can tell you very honestly that we couldn't have picked 2 worst years in the state of California to attempt to look at the effect of drought on the soil biota. We have rainout shelters, but that wasn't sufficient. Well, so we're looking at the flow of 13C carbon into the soil and where it goes, and using that to enable our metagenomics, metatranscriptomics, and particularly faunal and viral explorations. So I'm going to start talking about viruses and their role in controlling bacterial community composition and hopefully carbon flow near roots and near decomposing litter. There is much current interest in viruses in soil. This is a review that was published a little over a year ago with actually really nice pictures of the inhabitants. And we'll zero in here on a surface, on a mineral surface, with a water film and bacteria embedded within that water film, and phage, in theory, attacking them. So looking at those, the reason we are so interested in, the primary reason, not the only reason, is that it's pretty well thought, pretty well understood that in marine systems, phage are critically important in controlling the flow, the cycle of carbon, that up to half of the standing bacterial biomass is killed every day by phage attack, and that this turnover of carbon sustains the standing bacterial biomass in critical, well-studied parts of the ocean, such as the Chesapeake Bay. This is called the viral shunt, when phage attack the bacteria, releasing the bacterial cell sap, guts, soluble, readily usable carbon to be reconsumed by other bacteria. Well, depending on the efficiency you assume, a high percentage of that carbon will return to the atmosphere as CO2. A small percentage, perhaps 10 to 30%, will be refixed in bacterial biomass, the viral shunt. But also, we have the debris, the cell debris and the viral debris that is produced, and that material in marine systems settles to the bottom of the ocean and becomes a starting material for that huge organic pool of carbon in oceans. So, does this happen in soil? We do not know really the answer to that. Is it analogous to marine systems? Is there a phage shunt? We have a nice picture here of a bacteria, happy-looking bacteria being attacked by a phage, spilling its cell sap and being reutilized, much of it lost as CO2. And then that debris from the cell, or the viral particles itself, or the DOC, the dissolved organic carbon compounds coming out of the cell, could become involved in sorption to mineral surfaces or occlusion within aggregates. So that's what we're after. I'm going to start looking at the DNA viruses, particularly phage, using CRISPR-enabled metagenomics, direct extraction of VLPs, viral-like particles, and high-seq on those to get a virome. And then we'll go after eukaryotic viruses, particularly RNA viruses, using metatranscriptomic analysis over time. So this work I'm talking about, towards the end, I'll talk a little bit about the field data, but the upfront virus work is all greenhouse, carefully labeled stable isotope chambers. So we label our plants, we go in, we expose the soil, and we carefully excise the soil around the root, call that the rhizosphere soil. We then extract DNA and RNA from those roots, metabolites and proteins from that soil around the roots, from the soil around the roots. We really work in the rhizosphere of the soil, of the root. Okay. We then take the extracted DNA, run it through a density gradient centrifugation. I think we'll have several talks about that, which JGI has developed that capability as well to separate out the C13 labeled or the heavy DNA. And then we can begin the analysis of those materials. So in our lab, this was originally developed by Steve Blazewicz. Evan Starr has been analyzing the DNA from the rhizosphere soil. And this shows the brown trace is the unlabeled rhizosphere DNA, and the green trace is the 13CO2 labeled plant rhizosphere DNA. And in this early work, which was done now probably 5 or 6 years ago, he divided that up into two fractions. The heavy fraction, which doesn't overlap the C12 DNA at all, and what we call the middle fraction, which does overlap, but we can separate it from the C12 DNA in some ways by subtraction. So this simply shows the top 40 bacteria that we found in the heavy fractions. And it shows a couple of other things. One, it shows, actually, we're better, this a slightly old slide, but the little green circles on the top mean that those genomes are more than 90% complete based on single copy gene abundance. The blue circles are better than 70%, and the fragments are better than 50%. So we're beginning, in fact, we can reconstruct a much higher percentage actually right now. But our question isn't, can we find the bacteria who are eating the root carbon? Our question is, can we find the phage that are attacking the bacteria in the rhizosphere that are eating the root carbon? And if in fact they are living off of that bacterial carbon, those phage will be C13 labeled as well. So we go into that CIP fractions, the heavy fractions, and look for the C13 labeled phage DNA. And this shows 80 high confidence, mostly complete viral genomes, and their predicted host taxonomy. So we're able to find phage, we're able to a pretty good degree, identify the bacterial host. We're going to focus a little bit on the most abundant bacteria here, Burkholderia, which has basically a complete bacterial genome. And I realize this this bacterial genome is running off the side in terms of single copy genes and multiple copy genes. But this is the most abundant rhizosphere dweller, and it happens to have a CRISPR-Cas system. And I will say that very few of the bacteria that we find in soil have CRISPR-Cas systems. So we can identify the phage of this nearly complete genome. We can circularize the phage, and we can predict proteins, one of which has been confirmed by the proteomics group led by Mary Lipton up at PNNL. So that's one approach, which is CIP-enabled metagenomics. And this is a second approach that Alexa, a graduate student in my lab, is using, which is extracting particles from soil, taking the suspension, concentrating it, and you can either continue to purify and treat it and do shotgun sequencing, which is where I'll start, such that we have two types of viral metagenomes. We have the virome, that which we get from high-seq of viral-like particles, or we have the microbial metagenome with the viral scaffolds mined from that. And I'm not going to show the picture here that Alexa is supposed to make me on Monday. That's why you won't see it. But it shows that the viral metagenome looks like it in a diagram, looks like about this. And what we can find from the total microbial metagenome is a small subset, about 10% of the total viral metagenome we can find in our microbial CIP-enabled metagenome. There are a few viruses in the virome that do not appear in the soil metagenome. So this has interesting, I think, ecological implications about whether those viruses we're picking up in the soil metagenome are in fact active, replicating, and occurring as free virus or free viral particles in the soil. We believe, in fact, that most of them are, apparently. When we started this, we really didn't know for sure whether the viruses we were pulling out of the metagenomes were prophage or actually lytic and occurring as free viruses. This is something we do know now. This is Alexa's work. The gray part of the fan is previously identified viral sequences. The purple part is our new viral sequences. But every time you see data on viruses from soil, this is what it looks like. It's like a huge, we have a joke about this, huge diversity of never before seen viruses, yes. Okay, so we can sequence it, or we can make a right turn here and do imaging and see what they look like. So we've been working with Livermore to do nanosims, nanosecondary ion mass spec, to visualize the C13 occurring. And this is a nanosims of the viral-like particles from the plants that were 13CO2 labeled, and this is from the plants that were not 13CO2 labeled. So we're beginning to have a handle on the enrichment of the viral-like particles, which is a step towards establishing their importance in the carbon flow. Alexa's assignment is an easy, reliable enumeration of virus from soil and the ability to do the viral metagenomes. So those are all DNA-based viruses. That's what we're doing, HiSeq for DNA. But we were also interested in RNA viruses because many of the viruses that attack eukaryotes are RNA viruses. And this is work that was originally, the experiment that was originally done by Aaron Nuccio. And what this shows is that a little sidecar on the bottom there that the roots grow into. And we mark when the roots get to an area of soil, so we're able to come back and sample that soil 3 days after the roots get there, 6 days after the roots get there, 12 and 22 days after the roots get there. So the time frame of this sampling has to do with how long the soil and its microbes have seen an active root, a fresh young root. 4 sampling times. We have soil with no roots and no root debris. We have soil with just roots, that's rhizosphere. We have soil with just decomposing plant litter, that's just root debris. And then we have soil with the rhizosphere and the root debris. So there's 40, this ultimately, and 4 reps. Totals 48 reps. So that was the database that Evan Starr took to mine the RNA-based viruses, and looking specifically for eukaryotic viruses. And he identified them by their definitive RNA-dependent RNA polymerases. We found over 1,000 RNA viruses that affect bacteria. I'll talk a little bit more about those. And about twice that many RNA viruses that appear to infect eukaryotic organisms. The primary hosts for these RNA-based viruses in soil we believe to be fungi and a group of bacteria within the proteobacteria. If you're interested in this, this paper is uploaded on the bioarchives. This shows the phylogenetic fans, or trees, of RNA-dependent RNA polymerases. We, of course, get a high diversity of viruses that have never been seen before, spanning much of the known diversity of the RNA viruses. If we focus in on one group, we can see this is the group of RNA viruses that attack bacterial hosts, the gamma proteobacteria and alpha proteobacteria. And you can see the pink, again, are the sequences that have been seen before, and you can see that we can fan it out quite a distance of RNA viruses that have not been seen before. So, fungi seem to be a very important host for this newly reported group of RNA viruses. These groups are currently thought to only infect fungi in soil. We can also look a bit at the community ecology of these RNA viruses. And what this shows is the blue dots and squares are soil with no debris, root debris. The red dots and squares are soil with decomposing litter. And you can see that the primary factor that separates the group of RNA viruses is the presence or the absence of decomposing litter, which surprised us because in all of our other work, everything else has always been the presence and the absence of the rhizosphere. In fact, you are able, if we put in the rhizosphere versus bulk, in the blue group, you could see separation of the rhizosphere and the non-rhizosphere group until you remove the bacteria. And once you remove the bacterial viruses, they just mush together. So it's the bacterial response to the rhizosphere that you're seeing echoed in the RNA virus patterns. Here, this pattern, this response to root debris, we believe reflects the impact of fungi on the RNA viruses and the fact that fungi are the superstars of litter decomposition in soil, okay? So we are also very interested in, in the eukaryotes that we could find using RNA-seq. On the right-hand side, well, our amplicon of choice was cytochrome oxidase 1, which is the first column, with a lot of amoeba, fungi, and other eukaryotes. But the cytochrome oxidase database is weak at best. I mean, it's hard to classify things based on that database. And so we also went back and classified them. You'll see that the database based on 18S RNA is somewhat different, the abundances, than we get from cytochrome oxidase. We believe that's due to the fact that cytochrome oxidase is very sensitive to how active the cells are and how actively growing they are. Okay, so we have found if we look at the composition over time for 3 days, 6 days, 12 days, and 22 days, we find huge shifts in the composition of the viruses. Now, this could be due to dynamics of their hosts, but it is also likely due to attack, colonization, and lysis of the eukaryotic and prokaryotic cells. We find that the host communities are partitioning depending on whether the carbon they are using is occurring near roots or near decomposing debris. And the viral community, the RNA-based viral community, is based, it appears to be primarily on the presence of decomposing litter and not particularly on the basis of an active plant root. So, going back to our SIP enabled exploration of the rhizosphere soil. This approach of looking for bacterial cells that are heavily labeled, that is, are living off of root carbon, has allowed us to find interesting groups of organisms, one of which here, the purple group, is a TM7. And we've isolated and circularized that genome. It's a Sacar bacteria from the TM7 group. It's a small genome. Its GC content is low, and we believe it is likely an epibiont, perhaps an epibiont of an actinobacteria. It is from the candidate phyla radiation, but it is, I won't say that. Somebody asked me the day before yesterday if that is the one that was named after me. And the answer is no, it isn't. I wish it was, but that's what's obviously we call it Saccharomyces rhizosphere, yes. So there it is in the candidate phyla radiation. We can get, by looking at the genes present on the genome, we can predict metabolic characteristics, carbon utilization, energy generation, and this unusually for candidate phyla radiation appears to have the capacity to utilize oxygen, which is interesting. And we can also predict its potential for some of the interactions we're interested in, particularly response to antibiotics, capacity, sensitivity to antibiotics, potential interactions with plant hormones. So we can explore the rhizosphere players, finding previously unexplored biotic components, and we can propose, or at least hypothesize, the types of metabolisms they have based on their genes and the types of interactions that they are working on. So moving on to the bigger guys, the fauna. Who's there and how are they impacting the carbon? Fauna in soil are the major grazers of bacteria, and this has been known and studied in some detail, particularly in Europe over the last 20 years. It's pretty well established that faunal grazing can control the composition of bacterial communities and absolutely liberates the nitrogen that is tied up in the bacterial biomass. They should also redirect a substantial portion of that root-derived carbon towards mineralization. This work has been done and developed by Javier Ceja Navarro up at Lawrence Berkeley National Lab, who has used standard techniques to isolate this group of microeukaryotes, the aquatic organisms, the metazoa, and the nematodes, as well as the guys we're interested in the acari, the mites, the columbella, the small crawlers, I'll call them. And we can do this, particularly, we've been working on this in our 13CO2 field labeled soil, because we're concerned that the soil that we are using in greenhouse studies is not a good representation of the faunal communities present in soil. That soil has been sieved, it's been packed. It's very unlikely that the larger components are active. Hopefully, we can see a few things about protozoa and nematodes in the greenhouse, but clearly the field samples are the better approach. So Javier has used what I would call standard techniques to begin with, isolating nematodes and protists. He then uses a barrier, isopycnic barrier to centrifuge the soil through, leaving the cells suspended above that barrier. And some days, that is actually, you can see the organisms, the populations pooled and concentrated. And we can use those isolated faunal cells to extract DNA and to do metabarcoding. We also Javier has developed a high-throughput particle sorting approach to pull out the nematodes and the protozoa. We can then go into those groups of organisms that have been sorted and specifically use them for stable isotope analysis, for nanosims, and for sequencing. This shows a SEM on the right of a nematode, and the upper right-hand nematode, the little inset shows the nanosim. So we have a very hot, as in high concentration of C13 nematode coming out of that rhizosphere who has been actively grazing on the bacteria that have been eating the root exudates. So Javier has characterized the nematodes and the protist populations over time in our field samples. And the composition is not surprising, but it's using the primers to identify these is really an emerging method over the last 5 years, as opposed to the extraordinarily labor-intensive visual identification and picking. We've found, using qPCR of these primers, that we get pretty similar results on the right-hand side based on the primer qPCR versus direct counting, the labor-intensive direct counting in 3 zones of soil, bulk soil, near the roots, and actually on the roots. So it appears that our primer-based approaches are accurate in reflecting the who's there. Javier also isolates what we call arthropods. Those are the bigger guys, using a very traditional Berlese funnel approach, and we can get the organisms you get out of soil with the Berlese funnel. We can then, on individuals, extract the DNA, barcode it, establish the size of that organism, its metabolic rate, and we are developing our own arthropod database, microarthropod database, as well as protist database. Yes. We can also then take those organisms, there's one right in the middle of that pot, and sort them by morphotype, and then test the 13C content by IRMS. So if we look at the arthropod development or dynamics during the development of our plants, we find a very dynamic population over a period of 12 weeks. These are annual grasses that have a relatively short time frame in our California growing season, which is just ending. The samples were collected from next to the plant, DNA extracted for sequencing, and so we can characterize arthropods and protists now based on amplicon sequencing, which is a very nice development of Javier's work in the last 2 to 3 years. We can also find some of these organisms based on isotope-enabled metagenomics, but very, very few. In this case, it shows 2 nematodes that we were able to assemble from the 18S, 2 nematodes out of probably, I don't know how many, but a huge number of them. And we are able to get a little bit of genomic information from the isotope-enabled metagenomes. But it's very hard to get any eukaryotic information out of DNA-based metagenomics. So moving on to arbuscular mycorrhizal fungi and how they are interacting with bacteria and fungi and how they mediate the flux and fade of root carbon. So I'm going to talk here a little bit about our use of network analysis to identify modules of arbuscular mycorrhizal fungi, covariation with bacteria and fungi. And these are co-occurrence and co-exclusion groups, and they provide a starting point for understanding and hypothesizing the interactions that we believe occur between the AMF fungal hyphae and bacteria. So I'm going to start simple and use network analysis to look at bacterial interactions, and then ultimately AMF bacterial interactions. So this was the experiment that we're analyzing. It is our Avena barbata grown in our field hopland soil. Over time, week 0 up to 12 weeks when the plant is beginning to senesce. That's called, season 1. Then, after the plant has senesced, we dry the soil out with the decomposing root litter in it, simulating a Mediterranean dry summer. We do have dry seasons here, and they should start pretty soon, too. And then replant that dead rhizosphere and follow it over a second season. So let's see what we have. This shows the bacterial networks, that is covariation, co-occurrence, and co-exclusion networks over time. And at the first far left one is the bulk soil. Each little colored dot is a node that is a taxa that significantly co-varies with another taxa. And if you look across the top, we begin in the bulk soil to get some development of covariation patterns, but nothing like the pattern of covariation development, complexity, and amount that occurs in the rhizosphere soil, which is shown across the bottom. So in the rhizosphere soil, we find co-occurrence patterns of bacteria, some of which modules are quite large. That blue module there, by 12 weeks, has a number of members. And so we were very interested in pulling apart who was involved in that module of co-occurrence. And when we pulled those organisms apart and looked at what is called the module hub, and that is the taxa to which most of the other OTUs are significantly co-occurring, the hub of that module, we found that it was an organism that we thought we had seen before, that a former graduate student, Kristen DeAngelis, a long time ago, had isolated out of this hopland soil. It was almost 99% similar by 16S ribosomal RNA. We know that makes it similar, but certainly not necessarily the same organism. But it was similar to a rhizobium species that uses homoserine lactones for quorum sensing communication. We then looked at the other taxa, and half of the other taxa present in this module were alpha proteobacteria, which is where you tend to find the homoserine lactone quorum sensing. And of those, we knew that 24 of the 49 that we could identify were more than 97% similar to quorum sensing isolates that Kristen had also characterized in her earlier study. So this has led to a hypothesis. It's not proven, but it's a good starting point for a hypothesis that quorum sensing is a basis for co-occurrence patterns in that module. So what we were after here was exploration of the fungal bacterial co-occurrence or covariation patterns. And to do that, we looked at the co-occurrence patterns of ITS2 and 16S taxa. So this is all still amplicon based. And this is a little hard to see, but the top is season 1, the top two rows, bulk on the left, again progressing over time. The top is the bulk soil. The next row down is the rhizosphere in comparison. It looks pretty much like the bacterial co-occurrence patterns. And the bottom is the second season. And partway through the second season, we are getting into pretty dense and complex modules. So we can see co-occurrence patterns between bacteria and fungi that look somewhat similar. There's higher network complexity in the rhizosphere. It appears that the network complexity increases as the plant grows, and is pretty consistent with the bacterial co-occurrence network. But what we were particularly interested in was the interactions of AMF with the bacteria in this system. We know that AMF are supposed to interact with bacteria in soil in their hyposphere. We also know that they are responsible for moving a substantial amount of carbon from the root out into the surrounding soil. And so over the 12-week experiment, we were able to identify 131 distinct taxa that classified in the old Glomeromycota phyla, which was the traditional phyla that contained arbuscular mycorrhizal fungi by ITS2. And of these 131, we found 22 of those AMF taxa that were players or occurring in networks. And so this, particularly the paraglomus at the bottom and the glomus at the top. But, okay, and so we're able, actually, we know quite a bit about the biology of some of these AMF. Do succession patterns of the AMF bacteria and AMF fungal networks look similar to those of bacteria and fungal bacteria? I'll give you a hint. The answer is no, they don't. So you'll see these are much, much similar because we're much, much smaller and simpler networks because we've got a total of 31 OTUs that we can identify. If you look on the bottom left, you see the red dot in the middle is the AMF, the top triangle is the bacteria, and the bottom circle identifies the fungi, all in sufficiently small print that you can't possibly read it if you're interested. Hopefully we'll get this paper out within about a month. That may be optimistic. But we start, this is bulk soil. We start on the far left with a very simple co-occurrence pattern that slowly gets more and more complex until 12 weeks when it's starting to actually look like a network. This is the bulk soil. When we look at the rhizosphere soil, we expect to see a much more complex network structure, and what we find is not a single Glomeromycota node. We cannot find any Glomeromycota-based networks near the roots. If we look at the second season, which we have seen tends to be more complex, starting from the upper left down and back up to the lower right, we see this again is bulk soil. The development of reasonably complex networks based on co-occurrence patterns of bacteria and fungi with AMF. There are more networks or significant co-occurrence patterns of AMF and bacteria in bulk soil than in rhizosphere soil, and the complexity of these networks increased over time. We can hypothesize that the AMF bacterial interactions that result from interactions, physical contact, nutrient dependency, and we can identify several of the Glomeromycota taxa at several points within our network. We can see them moving through the network, if you will. So why do we, no, I'm not going to tell you. Why do we believe that the AMF co-occurrence patterns are occurring away from the roots? And it may be that our best explanation at this point, which seems to be quite accepted by AMF biologists and fungal biologists, is that the spores for the AMF, which are large, tend to occur away from the rhizosphere, and that the spores contain quite a bit of DNA for the AMF. There's maybe fewer spores in the rhizosphere. They may have germinated and are active as AMF. And as you know, AMF hyphae are odd when it comes to cross walls and nuclei. So it may be that there are very few occurring cell structures, cell nuclei in AMF hyphae that are active in soil. This is our best explanation of that pattern at this time. So we're also interested in imaging these AMF, and this is work done by Rachel Neurath, a graduate student in my lab. And I'm not going to explain the experiment except to say that she had little bags of minerals that were placed right on top of roots, of our Avena roots. And we then went, incubated those for about 3 months, and came back and looked at what microbiology was going on in those bags of minerals. And this is an SEM of a small diameter fungal hyphae that we found in the bag of kaolinite. And if we look in that little red box at the hyphae using scanning SEM, using nanoSIMs, nanosecondary ion mass spec, to show the C13, this is the hyphae with the kaolinite on the left, and it's a nice picture because it shows how the fungal hyphae can wear, if you will, kaolinite particles. So it's not that the bacteria is sorbing to the surface, but that the minerals, in this case, the kaolinite is sorbing to the surface of bacteria, to the fungi. And on the right side, we see very hot, very highly enriched C13. In fact, the C13 abundance in the hyphae is exactly what we find in the roots. So it is absolutely biotrophic, living off of the root carbon. The little blue splotches on that are basically where the kaolinite particles are blocking the visualization of the C13. So we can use stable isotopes to find the root carbon consumers and to map it into soil. We can use isotope-enabled genome-centric metagenomics to identify the microbial members in the rhizosphere or in the detritus or in the hyphosphere. We can establish their metabolic characteristics by analyzing those genomes. We can establish their carbon sources by supplying different carbon sources under different conditions. We can then use RNA-seq metatranscriptomic analysis to find eukaryotes, to find RNA-based viruses. And then all of this data goes into our network analysis and our food web models, which I'm not going to present any more of here, to identify and quantify the flow of carbon. So our exploration of this myriad of interactions is progressing very nicely, but in all honesty, it still has a long way to go. So 4 to 5 years, I'll come back and give you the whole story. The first authors on this are, about half of them have moved on to bigger and better lives, if you will, beyond UCB. Many of them are at national labs or Shenjings in New Zealand. The PIs on the project, JP, Jennifer Petrich at Lawrence Livermore National Lab, is instrumental in the isotope work, the nanosims, and has built a robot in collaboration with JGI to handle the rather labor-intensive isotope stable isotope separation, the density gradient centrifugation. Javier is our faunal guy who has pushed, I believe, molecular identification of fauna to new heights, and I think this is going to be key in our understanding of the role of fauna in these groups. Joe Zhao we're working with on network analysis, and Mary Lipton has been doing the proteomic analysis. We could not be doing the metagenomic, genome-centric metagenomic analysis that we're doing without our collaboration with Jill Banfield's group. And Angela Hodge has been our invaluable advisor, sometimes in-house. She has done a sabbatical with us on our vascular mycorrhizal fungi. So that's the story that I'm telling today.
So that was great. I have two short questions. One relates to vertical transmission of the microbiome that you're seeing. Could you provide some details on how you're treating the soil where you're planting these plants, and whether you have tried to look on the new seeds coming from these C13 labeled plants, whether there is new microbiome that is being vertically transmitted? And the second question is more of a curiosity, because normally the common understanding is that mycoviruses are transmitted only by cell-cell fusion, yep, and that the viral-like particles are not really active in infecting new fungi. So could you comment on that?
Since I remember the second question best, let's start there. Yes, the mycoviruses, particularly the mycoviruses, which are very small and... Sorry. Particularly the fungal viruses, RNA viruses, tend to be transmitted by cell division, by parental pass down. Yes, this is true. At least the primary group of fungal viruses that we identified, which are very, very small fragments of RNA, I think it's highly unlikely we would find those in viral-like particles free. I agree with you completely. Probably the only way we're going to find those is to look at metatranscriptomes of the fungi themselves, which is what we were doing, basically. Did that answer that question too? Was there another part? Okay. And then, well, how did we treat the soil? And how did we treat the seeds for our plant studies? So we've got an attitude, and that is that if you're interested in who over time has evolved to be a high capacity rhizosphere organism, you need to use soils that have grown that plant for 100 years, and plants that are common in that soil. So we generally collect our seed from our field site for the Avena, and the soil we are using, we take from our field sites that we know are growing significant number. It's not a pure culture, but a significant number of in our Hopland site, Avena barbata. So we assume that we are putting back together a plant soil microbial grouping that has been working together for decades. And so that's what we're trying to do. Are there bacteria carried on the seed? I would be surprised if there weren't, but we haven't looked at that. It's an interesting question.
A great talk. So my question, so you have these bacteria, the fungi, that arose in these microbial community interactions. I was wondering, in terms of soil carbon nutrient cycling, so could you comment on how important are these, different microbe communities? Like bacteria are more important, or others are important? And how might scale up from these microbial community to these local and global ecosystems.
So which of these groups of organisms is important in carbon stabilization and carbon flow? Okay. I think they're all very important in carbon flow. Whose components, whose bodies, whose cell sap are getting involved in stabilization processes? I don't know. There's a couple papers out suggesting and identifying certain types of cellular compounds, lipids. We're looking at EPS, extracellular polysaccharides, and how these become involved in absorption to mineral surfaces. But the group that gets involved in, basically, when you see the action of that kaolinite on an AMF hyphae, you can just imagine that hyphae being a nucleation zone for an aggregate, for the aggregation of soil particles. So there is a little bit of data on that. That we actually have a couple of experiments ongoing that are explicitly asking whose cell carbon is most important in being stabilized in our soil. Yeah, that's a good question. Yeah. Okay. One more?
Sorry,
One more
over
question?
here. Over on this side. Other side. There we go.
Other side, okay. Oh, there we go.
I was curious about your second season networks with AMF. So you found that the networks got more complex much more quickly.
Yes.
Do you have an idea as to why that happened, since the AMF should have died off, gone dormant, with the loss of the host?
Okay, so if you looked at that carefully, you found that at T0, at the beginning, before the plants were even growing, there was a relatively complicated AMF bacterial network. Well, that also fits with our starting hypothesis that those may be bacteria associated with AMF spores. Now, certainly at T0, there's spores there. Certainly at T0, there are not active roots or roots that are old enough to support AMF hyphae. So that's also another, yes. Clearly those interactions, those co-occurrence patterns were there from the very beginning. So that's another piece of evidence for involvement of the AMF spore. Yeah.